Okay, hello everyone. Welcome to this presentation on the anterior-posterior morphogen gradients in Drosophila. So hopefully this image gives you a little bit of a hint about what that means. You can already see a bit of a gradient of some kind there on the left side of the picture. So later on, we'll see exactly what this image is revealing. For now, I'll just give a quick outline of this presentation. We'll first review a little bit about what morphogens and morphogen gradients are. We'll talk about morphogen thresholds as well. Then we'll take a closer look at the specific morphogen gradients in Drosophila embryos, which organize the embryo from both the anterior and the posterior ends. We'll see how these gradients allow segmentation to develop. And then we'll take a look at the process of axis determination in flies and how that is similar to that in vertebrates. Now this presentation is mostly based on chapter 9 of the textbook, but a few other sources as well. And it's also not so much related to the video from Michael Baresi for this week. So you may want to watch that first, as I did. But I'll quickly recap what the main messages in that one were. So we learned that morphogens are molecules that generate patterns in tissues when they are distributed in a non-uniform way. That is, when they are in higher concentrations in certain areas of the tissue. When they interact with receptor proteins on cells, we can call them ligands. Cells that are closer to the source of the morphogen experience more ligand receptor binding for a longer time. So the difference in this signal strength contributes to the development of different cell fates in the embryo. Morphogen gradients can be altered in various ways. For example, by the extracellular matrix, the ECM, physically blocking the diffusion of the morphogen throughout a field of cells from its origin, or they can be altered chemically by antagonists, anything that would bind to a morphogen molecule and inhibit them from activating receptors on the cell. So those are the main messages of the video from Baresi. I would recommend watching that first, or at least soon after this presentation. Okay, so a concentration gradient is a continuous distribution of a morphogen. That means that there's a different concentration at each point along the curve, or at each distance away from the source of the morphogen in the field of cells. But not every value of concentration for a given morphogen results in a different type of cell. Then we would have hundreds or thousands of different types of cells all along the gradient from the morphogen. Instead, specific thresholds determine which genes are activated and which are inhibited by the morphogen. So in the figure, we can see that more morphogen generally equals more genes activated. A moderate amount of morphogen results in a different set of genes, likely a smaller set being activated, and a low amount of morphogen means even a smaller set, or perhaps no gene activation at all. So to be considered a morphogen, a signaling molecule must specify at least 2 thresholds, like this one in the figure, but some have been identified to have up to 7 distinct thresholds of gene expression. And when the theory of morphogens was being described initially, before there was experimental evidence of it, it was theorized that they could go up to 30 different thresholds, meaning they would specify 30 different sets of genes based on different concentrations. Now, we haven't found any morphogen patterns that go up to that level of specificity. Okay, so this figure is figure 4.23 in the textbook, and it reveals an experiment that was performed by different researchers in 1994 and in 1998. And it shows the threshold theory from the previous slide in action. So these researchers they took beads containing different levels of activin, which is a morphogenic protein, and they placed these beads in the middle of a cultured field of unspecified amphibian cells. So as these beads released the activin over time, it diffused across the cells, decreasing in concentration as it went. The concentrations of activin closer to 4 nanomolars activated enough receptors on the cells to introduce the expression of the Bicoid gene. So that's right over here, the dark red area. Concentrations at lower amounts only activated this XBRA gene. And now the very sharp boundary between these two regions is enhanced by the way that the protein product of XBRA actually inhibits goosecoid transcription. So there has to be a lot more morphogen or activin present in order to overcome that inhibition by XBRA and actually allow this other gene goosecoid to be expressed. So this experiment also shows how activin is a paracrine factor. It can diffuse across cells up to a certain range, and it affects all the cells within that area. So, and as that video from Berezi earlier explains, this is unlike a juxtacrine factor, which is transferred between adjacent cells on a more one-to-one basis. Okay, so morphogen gradients can occur in the syncytial blastoderm. So before gastrulation, which we learned about in the previous presentation, Drosophila embryos, like all insects, consist of syncytial blastoderm, like one big cell with many nuclei. The nuclei are all held in place by cytoskeletal elements branching out from each nucleus, and figure A shows this in an actual embryo and with a diagram. So they have these fibers of cytoskeleton that all interact with each other and hold the nuclei in one place relative to another. Now, because the nuclei aren't constantly floating freely around in the embryo, this means that the nuclei can experience different concentrations of morphogens when those morphogens diffuse from a point source. If they were all floating around all the time, then the morphogen gradient wouldn't be able to be established because the amount of morphogen each nucleus would encounter would be completely random. So figure D shows gradients of two morphogen proteins, bicoid and caudal, which we will take a look at in a moment. It shows how the location of the sources of these proteins contributes to the differentiation of these nuclei, which creates an anterior to posterior axis. So once cellularization occurs and the nuclei of the syncytium become segregated into their own cellular membranes, this anterior-posterior axis remains. We can see from the figure that the nuclei that are exposed to lots of bicoid go on to produce the head, and those exposed to lots of caudal go on to produce the tail. And the reason that this occurs is that these morphogens are able to activate different numbers of genes at different concentrations, which we just saw. Okay, so bicoid is the morphogen whose gradient is responsible for the development of the anterior end of the Drosophila embryo. Now in Drosophila oocytes, egg cells, the bicoid gene is already localized at the anterior end of the oocyte, but it's not actually translated until fertilization begins and the zygote begins developing. So this figure, which is 9.10 in the textbook, describes an experiment performed in 1998 and 1990 to determine the role of bicoid. So researchers located the mRNA for the gene bicoid in the anterior ends of wild-type embryos, and specifically in the acron, which is the small region of cells at the very tip of the anterior end. So they found that embryos with a mutation that caused a deficiency in this gene, the bicoid gene, failed to develop not just a head, but a thorax as well. Likewise, they also found that wherever they added bicoid mRNA, a new head region was formed, flanked on either side by thoraxes. And this follows the theory of morphogen gradients that we just described. So a high concentration of bicoid protein is necessary to turn on the genes responsible for the most anterior structures of the head. A moderate concentration is needed for the rest of the head and the thorax, and the specification of the abdomen requires a low concentration of bicoid. And you can see that where they added the bicoid mRNA to the middle of the mutant, it didn't just produce a thorax in one direction, it produced a thorax on all of the sides of the head. Now, there are, of course, multiple more specific thresholds for the multitude of genes in each of these regions of the embryo, but it does kind of follow that high, medium, and low concentration gradient. So this experiment was hugely important in developmental biology because it was the first time that researchers actually proved the existence and the role of any morphogen gradient, not just in flies. Before that, it was just the best way to explain how the axes in the embryos are formed. And the theory behind morphogen gradients went back to actually the beginning of the 1900s, but no experimental evidence for it was found until about 30 years ago. So at the same time as that previous experiment, researchers were visually demonstrating that the bicoid protein was indeed produced in the acron, and it diffused across the embryo towards the telson, which is the other terminal region. Now this figure, 9.14 in the textbook, shows a Drosophila embryo stained for bicoid, the protein this time, not the mRNA. And you can see that it's much more concentrated in the anterior region, and it drops off rather sharply. And you can also see that unlike the image on the cover slide that was stained for bicoid mRNA, this image, stained for bicoid protein, shows that the protein is most concentrated in the actual nuclei. The mRNA would be what is actually diffusing across the entire syncytium, but the proteins would only be produced and concentrated in the nuclei at this point. And the graph below shows you that indeed the concentration drops off steeply at first and then slowly later on. It also shows that mutants for bicoid don't have this gradient effect. They just have a small amount of the protein throughout the entire syncytium. So what does bicoid actually do once it's diffused in a gradient across the embryo? So for one, it acts as a direct transcriptional activator, which is a transcription factor that binds to and induces expression of a gene. And what it does is it targets genes that control body segmentation, like a certain important gene in Drosophila called hunchback. Now, by activating this gene, it creates a hunchback gradient similar to the existing bicoid gradient. Hunchback, on the other hand, activates gap genes, among other genes. So in insects, gap genes are a class of genes that are responsible for the development of the segmented body plan. And they're called that because mutations in these gap genes create a gap in the adult fly, or the larva, as if one or more segments were actually missing from the body. So the bicoid and hunchback genes work together to induce the expression of these gap genes and of other groups of genes. So bicoid also acts as a translational repressor, as well as a transcriptional activator. And translational repressor being a molecule that blocks the translation of a protein by binding to its messenger RNA. And it represses transcription factors that are normally activated by posterior morphogens. So bicoid being an anterior morphogen, it not only activates anterior genes, but it also represses posterior morphogens as well. In this diagram, one of these is caudal. You can see that labeled as CAD, bicoid being BCD. Bicoid, this red blunt arrow means it directly inhibits the translation of caudal. Now this diagram, which is figure 9.17 in the textbook, it shows you how many of these genes interact with each other and how they are all regulated by the bicoid gradient. So at the top you have bicoid. You can see that it directly activates three of these other, these four important abdominal gap genes, NERPs, KNI, hunchback, HB, and giant, GT. These all, on the other hand, regulate each other as well. You can see that bicoid activates the hunchback gene, and the hunchback protein both inhibits and activates another gene, Krupel, which is KR. So in the rest of the figure in the textbook, it expands on this, and you'll see that the hunchback protein, when it's at high concentrations, inhibits Krupel expression. But at moderate concentrations, that actually activates it. So you can see how a bicoid gradient would induce a hunchback gradient, which would create different levels of expression of this other gene, crouple, at different locations along the embryo, along that gradient. At the bottom of the diagram, we have a gene called tailless, TLL, named because flies without it have no tails. This is a posterior gap gene that inhibits all of these abdominal gap genes when it's expressed enough. So if we imagine a fly embryo sort of overlaid on this diagram, we can see the main message. So the bicoid gradient, which originates from the anterior end, regulates a whole bunch of other genes in the head, the thorax, and the abdomen that are necessary for segmentation. At the same time, posterior gap genes are trying to inhibit the expression of these genes. The morphogen with a higher concentration will win out, and we get segmentation where we expect it, more so in the middle of the fly. Okay, so Gilbert and Baresi identify 4 important protein gradients in the early Drosophila embryo, and we just looked at the first 2, bicoid and hunchback, which both originate from the anterior end. And we saw that the gradient in bicoid also produces a similar gradient in hunchback. Now we'll take a look at the other side of the embryo and the 2 main posterior morphogens, which are nanos and caudal proteins. Now this figure, figure 9.13, shows us how the concentrations of all of these proteins and their associated mRNAs change between their location in the oocyte and in the embryo once the oocyte's fertilized. So in the oocyte, this part A of the figure above, we can see that bicoid and nanos mRNAs are almost entirely located in the anterior and the posterior regions respectively. Meanwhile, hunchback and caudal mRNAs are found all throughout the oocyte. They haven't been localized into any particular region. Now, once fertilization occurs and the oocyte becomes a full zygote, bicoid and nanos begin to be expressed. And as we just saw, the hunchback protein gradient is induced by its activation by bicoid and its inhibition by posterior morphogens. So a similar thing happens with the nanos and caudal proteins. You don't get the gradients in hunchback and caudal until the gradients of bicoid and nanos are both established. Now, one primary role of nanos is to block the translation of hunchback mRNA, since we don't want any midsections forming in the tail end of the fly. So caudal protein activates genes that form posterior structures, just like what we saw with hunchback earlier. So nanos acts translationally, and caudal acts transcriptionally. But they both work together to activate and repress the correct genes at the right point. So at this point, I'd also like to remind everyone that all of this is still happening in the syncytium. There are no cell membranes to get in the way of the diffusion of any of these proteins. All of the gene activation and inhibition is occurring just within the many nuclei within the cytoplasm of the entire embryo. So now that the anterior-posterior axis has been established in the Drosophila embryo, it can be used to create the segmented body plan that is characteristic of the larva and the adult. So we learned earlier about the processes of specification and determination. At this point in the development of the embryo, the cells have been specified by the morphogen gradients, but this specification is somewhat flexible. It can still be changed by signals from other cells. Determination is the next step in the commitment to a particular cell fate, and this step is not reversible. So we've already talked about the gap genes. These organize the embryo into parasegments, which are so called because they consist of the front portion of one adult segment and the back portion of another. So when researchers were analyzing these genes, they found out that it appears that parasegments are how the embryo is organized, and the actual segments become clear in later developmental stages. But it's not so much as important as realizing that there's just different ways that the embryo organizes cells related to how those cells are organized in the adult. So the gap genes also activate another set of segmentation genes called the pair rule genes. And these genes create further patterns within the segments, and they also serve to activate this third set called segment polarity genes. Now, the segment polarity genes are only expressed once the syncytium has divided into distinct cells, and their function is to finally determine the cell fates in each segment and actually define an anterior and posterior portion of each segment. And all of this is dependent on the original anterior-posterior axis that was set up by that initial bicoid gradient. Okay, so ever since morphogens were established as a means of patterning the early embryo, there has been a ton of research into morphogen gradients in several different organisms. Since the 1990s, there have been over 700 studies on these gradients. And they've all found that morphogen gradients as a design tool are very similar across different species and in different structures within those species. One striking similarity is that between the anterior-posterior gradient in flies and the dorsal-ventral gradient in the neural tubes of vertebrate embryos. So the neural tube is very different in structure to the Drosophila embryo. Instead of a syncytium with a bunch of nuclei that divide in a synchronous way, the neural tube is a sheet of pseudostratified epithelial cells that are all reproducing on their own. And then that sheet folds over on itself and becomes a tube. So for this reason, simple diffusion of morphogens is not the main way that the gradient is formed. The neural tube morphogen gradient relies on more complicated actions between morphogens and the proteins in the extracellular matrix, which wouldn't be an issue in the syncytium of the Drosophila embryos. Now, despite these major differences, the morphogen gradient concept works in a highly similar way. So if we take a look at the right side of these figures, we can see that instead of bicoid, a protein called sonic hedgehog that I've circled here in red is what initiates the creation of the morphogen gradient in the neural tube. And it's produced by the notochord, which lies just below the neural tube, and it emanates from the ventral side of the tube. It binds to a transmembrane receptor protein, which is pitch, labeled here, which stops the inhibition of another transmembrane protein, SMO, and that allows the production of a transcriptional activator group of proteins, which is labeled here as GLI-A, and instead of repressor proteins, GLI-R. So normally, the repressor proteins are all that is being produced. This pathway allows the activator proteins to be produced, which in turn repress the repressors. Then these transcription factors are present in a gradient because the activator of this whole pathway, sonic hedgehog, was also present in a gradient. And this gradient of transcription factors actually activates the target genes of the whole process. Now, this entire gradient is opposed by a gradient of two signaling molecules, which are here as BMP, or bone morphogenic protein, and WNT. And these are generated at the dorsal side of the neural tube. So just like in the Drosophila embryos, there are two gradients coming from opposite sides, and they both work to establish an axis. So taking a look at the right side of the figures, we can see that the target genes of these morphogens are all mutually repressive, which is shown by these blunt arrows in both directions. So they're all mutually repressive, just like in Drosophila and in the experiment we saw at the beginning of this presentation. And this mutual repression refines the patterns that these gradients create, and it creates sharp boundaries of gene expression. So between Drosophila and the neural tube, you can see that different proteins and different signaling pathways are at play, but they're all resulting in very similar results. Okay, so let's go over what we discussed in this presentation. We learned about the principles of morphogen gradients, which are all laid out and kind of summarized on this figure on the right. A morphogen is secreted from one or more cells at a specific location, and it spreads across cells to create a gradient of decreasing concentration. Morphogens signal cells to produce transcription factors, which are different depending on the concentration of the morphogen, that activate or repress target genes. These target genes then initiate networks of transcription factors that generate the actual pattern along the axis created by the gradient. We also looked at how thresholds are important for enabling morphogens to activate different genes at different concentration levels. We saw that in Drosophila, the anterior-posterior axis is determined by a gradient of the protein called Bicoid, which originates from the anterior, and it's opposed by a similar gradient originating from the posterior. This anterior-posterior axis is important for the development of segments later on, which will go to influence the entire body plan of the fly. And finally, we saw how axis formation in the fly embryos is very similar to that of neural tubes in vertebrates. Okay, so that's all. Thanks for watching. And I would recommend either looking at the textbook or watching that Baresi video afterwards. All right, see you all later.